our pressure flow model presentation to model the flow of sap throughout the phloem. And so we start over here at the source cell where we have glucose that we've retrieved from photosynthesis. And this glucose is used to synthesize these sucrose molecules. And so we no longer need these glucoses, and we have sucrose in the companion cell. So the companion cell is basically the cell that is in charge of loading and unloading solute from the phloem to source cell, or from the phloem into sink cells. And so this process for the source cell into the phloem requires active transport, which is why we created, and there's two ways in which active transport takes place. Either through the symplastic way, which is just these sucrose molecules move through these plasmodesmata from the source cell. Plasmodesmata is like the component connecting both the sucrose, both the phloem and the source and the companion cell. And the other way is the apoplastic, which requires this whole proton gradient. Protons are pumped out of the companion cells and back into it and returned via like a co-transport protein. And then this so when that happens, we use ATP again to move the sucrose into the phloem. So once this phloem is here, it creates a hypertonic solution because originally the phloem did not have any sort of solute in it. And so the water, that's what we acquired from via transpiration. And so if via mass flow, it moves through the xylem, and then it moves into the phloem via osmosis. So we now have our water here, and the water moves because we have a high solute concentration and not high water concentration. And so these little darker pink components are called sieves, which are basically just elements that allow things to move through the phloem, or I guess in this case down the phloem. And so when that moves, the water and sucrose both move down the sieve elements. And then from there, the sucrose is then transported back into the companion cells, and the water moves back into the xylem, because there's no longer high concentration here. And from there, but this whole process of like flow moving down is called translocation, which is basically actively loading solutes down sieve elements, and the xylem drawing back the water from the phloem. And so when the sucrose goes back into the companion cells, these companion cells actively unload the sucrose into the sink cells via, again, ATP. And then when these sucroses reach the sink cell, that is basically how this whole process ends. It's important to mention that this whole process can be reversed, where like mass flow can be reversed, and the direction can just change. And the pressure gradient from the phloem is what actually like allows all this to happen. The pressure gradient is caused by the transpiration and water flow, and it moves down and also moves it over here. And that concludes our presentation of pressure flow model.